Hello everyone. Let's continue our discussion on assisted reproductive technology, and in today's class we are going to understand something about artificial insemination. Now, what are the learning objectives of current study? To understand the principle of artificial insemination, to learn the procedure of artificial inseminations. The session outcome is understanding the procedure of artificial insemination. We'll try to understand as much as possible. Now, let's start our discussion. Artificial insemination. Now, what is this artificial insemination? It's a method of treating infertility by inserting semen directly into a womb. Insemination refers to adding or releasing semen. Remember, semen is a fluid, a mixture which contains spermatozoans. Okay, so releasing semen is called insemination, which usually occur during natural intercourse, but here it is done artificially. So this method of treating infertility. See, in the previous classes we have discussed about one of the method of treating infertility, that was IVF, in vitro fertilization technique and embryo transfer, simply called as test tube baby. Okay, so here in this particular method of treating infertility, semen is directly inserted into the womb of mother. The intrauterine insemination, artificial insemination, is also known as intrauterine insemination, and it is the most common method. Okay, and this artificial insemination can benefit couples or individuals with the range of needs. So it can help the following medical situations. In a woman, cervical factor infertility. So if there are certain reasons for infertility that is associated with the cervix of the uterus, remember the neck of uterus is called as cervix. So any problem associated with that portion of the uterus is called as cervical factor infertility. So in such cases, the cervix either does not produce mucus that helps the sperm to travel to the womb, okay, or mucus contains a substance that kills the sperms. In both these conditions, okay, the woman might be infertile, and those causes are called cervical factors. So this artificial insemination can help in bypassing this problem. Okay, these two problems. So main problem is in the neck itself. So spermatozoa must pass through the cervix to enter into the uterine cavity. So, but it doesn't happen because the mucus, its production is very less, or it might contain certain substances that might kill the spermatozoa. So to bypass these two problems, artificial insemination is done. I'll repeat, artificial insemination is the process of directly inserting the semen into the uterine cavity. Okay. Artificial insemination can also be successful in mild to moderate endometriosis. We have already discussed what is endometriosis. You must be familiar. So even in case of endometriosis, this can help. In rare cases, women may be might have allergic reactions to certain protein in sperm or in the semen. In very rare cases, one might be allergic. So in that case also, one can overcome that problem by artificial insemination. This intrauterine insemination, I for intra, U for uterine, second I is for insemination, can help to support the couple who have medical conditions like erectile dysfunction. If there is a erectile dysfunction in case of male, in that case also artificial insemination might help. In addition to that, it can also be useful in oligozoospermia. Oligo, you are familiar. Few. Zoospermia refers to very few spermatozoans. So, where the sperm count is less, in that case also artificial insemination can be useful. Or sometimes in certain cases they are non-motile. The spermatozoans are non-motile, so they won't be able to reach up to the uterine cavity. Condition is called as asthenozoospermia. In this condition also, IUI can be useful. Next is the, sometimes what happens, these medical treatments can can be a reason for infertile, such as radiation therapy. If someone is going for it, in that case also intrauterine insemination can be very useful one. Now, let's see what are the procedures, steps that are followed during artificial insemination or intrauterine insemination. First stage or step is obtaining the semen sample. Second one, preparing the semen sample. Third is implanting it into the uterus. So these three are the steps that are involved in intrauterine insemination. First, let's see how semen is obtained and prepared. So the sperm that is used in artificial insemination either can be provided by the husband himself or sometimes by donor too, anonymous donor. So the source of those spermatozoans or the semen will not be known. So if it is from a donor, then this will be supplied by a sperm bank. What is sperm bank? We'll be discussing in our next class. So from sperm bank, this semen sample can be taken. So in such cases, what they do is that they will quarantine the sample and also test for any transmittable diseases, because the person might be infected with certain diseases, and if such semen is used, there is a chance of risk of transmitting such diseases. So those sperm bank staff will make sure that none of those such kind of infections are passed from the donor to the one who is going for conceive. Okay. Next, here usually what they do is that the actual process of sperm bank we'll be discussing in the next class, so I won't stress much upon that. Let me continue with my discussion here. So here what they do usually is that the semen will be frozen, and then they'll process it properly, and before using it will be processed properly. But during the freezing process, that is during the storage, semen will be usually added with cryoprotectant. Certain chemicals will be added. Cryo usually refers to the lower temperature. At lower temperature, there is a chance that the cell might get damaged. So to protect that shrinkage of the cell or something like that, cryoprotectants are used. Okay, and in case if the partner, the husband or partner is donating his semen, then directly the samples can be taken, as a fresh sample can be taken. Now, how it is removed? Usually it can be removed, taken by different methods. So there is also one method of surgical sperm aspiration. In this case, what happens? A physician removes the sperm directly from the male reproductive tract. This is usually done in case of erectile dysfunction. So directly during in surgical sperm aspiration method, they'll remove the spermatozoans or the semen. So once the clinic has the sperm, they will wash it and remove any other elements that are like impurities that are present that might interfere with the fertilization. So any impurity that is present can be removed, so that only spermatozoon will be left in the sample. So that those are the first two methods, steps. First one is obtaining and then preparing it. Third one is implanting it into the uterus. So here what happens is the fertility doctor will implant the sperm directly into the uterus by using a fine catheter. Okay, so this is done by keeping the vagina open. Okay, and then the catheter will be passed through the cervix. If there was a problem in cervix region, this can be overcome. So through the cervix it can be passed, and the doctor pushes the sperm through the catheter. He'll release it into the uterine cavity. So here you can see the sperm collection, and it is then processed, and then insemination is done through the vagina, through the cervix. It is directly passed to the uterine cavity, which will then swim directly towards the fallopian tube where the fertilization takes place. Okay, and but in case this transmission is done between the partner, not only even in case of donors, doctors usually give ovulation prediction kit, because success rate is too high when the spermatozoa is released close to the ovulatory period. So by using this ovulatory prediction kit, one will know exactly when the ovulation is going to take place. In this case too, just by what we have seen in case of in vitro fertilization, the female can be induced by giving certain hormones like FSH, LH or human chorionic gonadotropin to improve or increase the number of ova or maturation of the follicles. Okay, so that on the day of ovulation, maximum number of eggs are available. Okay, and then on the same day, or at least a day prior to it, one can release this spermatozoa. So the next day or the other day after that, fertilization takes place, and in few days implantation might occur. So in summary, if we have to tell what exactly artificial insemination is, the first step is ovarian stimulation, where hormones are given so that more and more follicles are produced, released at least, and then usually ovulation induction is done. So like one might be, as we have discussed earlier, one might not be successful. So more than one matured follicles are needed to confirm this process, and this is done by human chorionic gonadotropin shots. So this will help in improved maturation and timely ovulation. And then sperm preparation we have seen previously too. So here semen sample is washed and by means of a sperm capacitation to remove any other additional contaminants that are present. So only motile spermatozoans are available. Next step is insemination, where again I am repeating it, by using catheter the spermatozoans are released. But remember, no anesthesia is required during this process. And just to make sure that after fertilization implantation occur and the uterus is ready to take up the blastocyst, progesterone is administered, and here it will be vaginally to boost the chances of embryo implantation. Remember, progesterone will also be given during in vitro fertilization, again for the confirmation of implantation, because we know progesterone is a pregnancy hormone. One of its function is to prepare the endometrium for future implants. Now, what are the risks of this artificial insemination? So, conceiving twins or triplets is more. Just in case of IVF, here too the chances of triplet is more, and this increases if they are using other fertility medication too. Like gonadotropins are given, if it is given to induce the ovulation or maturation of follicle, the number of follicles that are that are released during ovulation will be multiple. So triplets, twins, triplets or multiple births are common even in case of artificial insemination. And there were certain chances that there might be premature birth or miscarriage. So that happens when they have more than one fetus. So if there is only one fetus, chances of miscarriage are less. But if the number of fetus are more than one, there is a chance that the babies will be prematurely born or there are miscarriages. One more is, as we have seen in IVF, ovarian hyperstimulation syndrome, OHSS, can also be one of the condition where the ovaries will swell after combining fertility medication plus IUI. So triplets or ovarian hyperstimulation syndrome. This will be usually characterized by mild pain in the beginning, but after conception the pain might be chronic too, and it might continue for few more weeks. Okay, and one more very important thing is that after intrauterine insemination treatment, the woman should wait for 2 weeks, at least 2 weeks before taking the home pregnancy test. Nowadays home pregnancy tests are available, so that can be done, but after 2 weeks. Because what happens, hormones will not show in a test before that. Before that hormones will not be visible. So these kits are basically hormonal methods where the biosensors are used to detect the level of hormones in the blood. So usually in these kind of treatments, it takes minimum 2 weeks for the raise of such hormones in the blood. Detectable level of hormone will be raised only almost after 2 weeks. So one can confirm the pregnancy after 2 weeks. 3rd week one can confirm it, and then rest of the procedure is almost same like normal pregnancy. One should be always under the observation so that pregnancy period, gestation period is passed smoothly. Yeah, so that's all for today's class. Let's just see the summary. So artificial insemination is a method of treating infertility by inserting the semen directly into the womb, artificial. So it's not natural. Insemination is a technique of passing or inserting the semen. It can be natural or it can be artificial, but here we are talking about artificial, where it is done by certain means. Okay, and the steps involved in intrauterine insemination are obtaining the semen sample, preparing the semen sample, and implanting it to the uterus. And these are my references for today's class. Thank you.